Hi, everybody. Welcome to the 14th episode of the Labiotech Hangout. And so this month we have the co-founder of Tolremo here. Hi, Stefanie.
Hi there.
So Tolremo is one of the most promising biotech startups in Switzerland. I heard quite a lot about you, so it's great to have you here. And so you're based in Zurich, spin out of the ETH, and you're really focusing on preventing resistance development in oncology or cancer. Mm-hmm. And so you were founded March 2017, and lately you raised 8 million euro round, so 9 million francs in Series A in September 2018, which is actually a good job on that. Congrats. And maybe our first question is really on drug resistance, really interesting topic. And it's something we hear, I mean, more and more, we cover it more and more, especially hear it from companies developing drugs and the drugs get resistant. Mm-hmm. And so how do you plan to tackle it? Why do you believe you will be the good one to tackle it?
I think our approach is a little different. So usually when people think about drug resistance, they think about antibiotics and drug resistance. So everybody's aware that this is one of the major problems that we'll be facing as a society, basically, in the next couple of years to come. Cancer drug resistance, people are a little less aware of. And classically, when they think about drug resistance, they think of mutations that are acquired in tumors after a couple of months or even years of treatment. And these mutations are really then, at that point, drug resistance is really hardwired into the DNA of a tumor. It's really difficult to do anything about it then. Our approach is a little different. We target
just the hard resistance, you mean like after the second, third line of treatment, so like after 2 or 3 years when the tumor mutate and becomes resistant?
Yeah, it's continuing drug treatment is a selection pressure on the tumor. And so over time, and many studies have seen that over time, the tumor gets more and more diverse and different cell populations acquire different resistant-causing mutations. So you end up with an extremely complex drug-resistant tumor. And even if you did have an additional cancer drug that would target one of these resistance-causing mutations, you have all these other ones that you're not tackling.
Yep.
And so what we do is we want to prevent that we get to this stage to begin with. And 88, we found that before you actually have genetic drug resistance with these new mutations, we see that cancer drugs induce stress responses in cancer cells. And these are transcriptional responses. So here we're still talking about software in a cancer cell. And the nice thing about software is, of course, you can change it. You can change it before you have this hardwired hardware defect in the cancer cell.
Okay.
So we develop small molecules that can be combined with existing cancer drugs to prevent these escape software programs running in cancer cells.
When you talk about software, I mean, it's a good image, but what does it mean biologically? Is it the cell structure?
It's transcriptional responses in cells. So a broad panel of genes that cells express, they start expressing, and this helps them fight off the stress of being attacked by a cancer.
Yeah, and you measure it by looking at the cells, and you can just, I guess, a mix of sequencing, looking at the cells, imaging, and to kind of quantify that.
Yeah, right? Originally we set up so in the beginning, when it was still in academia, it was a new conceptual framework, really, how we think about drug resistance and how we can potentially target it. And then we developed a drug screening development platform that is image-based, that allows us to screen thousands of drug combinations. And that was our starting point to the company to develop our own chemical matter that prevents these transcriptional adaptations.
Okay, I see. And so basically, once you have this data, like the framework, then you can adapt the drug, or like the drug combination, tailor the drug combination to that. In theory.
In theory, yes. At the moment, we develop 2 assets in parallel. And the nice thing about it is that these transcriptional escape mechanisms, they are triggered by multiple different cancer drugs. So our add-ons can be combined with different cancer drugs, the very same add-on, different cancer drugs used for different cancer indications to boost their long-term efficacy.
Okay, I see. Okay, so that was one of my questions actually, from what kind of drugs you're developing. Is it, so you answered small molecule and combining with existing cancer drugs, I guess monoclonal antibodies mainly, I guess. Like,
it can be monoclonals, it can also be small molecules.
Okay.
It all depends on the mechanism of action that they have.
And what's the reason behind why don't you develop like your own monoclonal antibodies, or like more develop like cell therapy or whatever?
Now you're breaking up.
Why did you choose to do your own small molecules and combine with existing small molecules and monoclonal antibodies versus developing your own monoclonal antibody, or a more like, let's say, high-tech CAR-T or cell therapy, which seems to be very trending at the moment, or immunotherapy or checkpoint inhibitor or something in that direction?
Even there, we do see drug resistance emerging. Almost in solid tumors, drug resistance is really the limiting factor, what limits patient survival in the long term.
Yeah.
But the reason why we didn't go for monoclonal antibodies is small molecules are easier. When they're easier, we can have very, very fast cycles of different variations of these compounds. Preclinical development is much easier, much faster, and cheaper.
But the efficacy is, it's not comparable. If you compare a CAR-T or checkpoint inhibitor with a small molecule, it's
not necessarily.
No. You tell me.
No, I mean, it really depends. For some indications, we have the same mechanism of action where we use either small molecules or antibodies, really for the same purpose.
Yeah.
Immunotherapy, yes, we do see very durable responses in relatively small patient populations that unfortunately at the moment we still can't select for. But also in small molecule-based therapies, we have long-term survivors.
Yep.
What we work on, for example, is in BRAF inhibitors in melanoma, for example, we do have about 20% long-term survivors.
Yeah, okay.
So it is possible to have long-term survival, even with small molecule. And our goal is to push this up from the 20% long-term survivors to, in an ideal case, 100%.
Yeah, okay.
Eventually it's clearly done.
And I guess, I mean, my data is more like the CAR-T or Novartis CAR-T, which is more like
even
80% long-term survivors, at least in the phase 2B, but I don't know on the market how it looks like, but yeah.
And in the CAR-T, so their reimbursement
it's
model,
a very specific,
it has to work.
And it's a very specific indication as well, but
yeah. But even there, they say, I think people only have to pay for CAR-T once they see responses. But these responses, I think, are defined for your stable disease for 3 months. They're not going to say you only have to pay for it if it works for 2 years. That would be too risky for them. So they're limited quite a bit there.
But I mean, I'm talking about the study. I think it was over 3 years, and that over 80% of cancer-free over 3 years. I think it was over 3 years. So anyway, sounds good. And I want to pick on what you said on the limiting factor on solid tumors, because obviously on solid tumors, CAR-T doesn't work at all, or even cell therapy in general. I think checkpoint inhibitors, it's still not completely clear, or doesn't respond that well. So why do you say, you said on the solid tumors, the limiting factor is really the drug resistance? Can you expand a bit on that?
It's basically what I said.
Yeah.
Let's say, for example, or another example is non-small cell lung cancer. Have certain mutations, for example, EGFR mutations. We have very potent EGFR inhibitors that will inhibit this receptor. They work beautifully, and you see on PET scans, you see tumors almost melting away within weeks. but they come back usually within a year or so, patients would break through that again. And that's really what's limiting. It's not that there is a lack of available cancer drugs, right? We do get limited responses for patients, but what we have to do, we think we don't need yet another cancer drug on the market where we can have 1 year survival, 1 year progression-free survival. What we'd like to do is we take what is out there and we make it better.
Okay, so for you, basically, it's not the problem of available therapies, it's really like the drug resistance is more important than the available therapies, or than the combination with like patient or like operations or radiotherapy or whatever.
Yes. That's why in oncology we talk about first line and then second line and then third line. There are options. They're just not very durable.
Yeah, definitely. On the clinical stage, so you said you were in preclinical studies at the moment, or can you expand a bit on the clinical stage you're at?
Say again?
On the own drugs you're developing, what's the clinical stage you're at? It's preclinical, that's correct.
Preclinical, yes.
How's it going so far?
So far, it's going surprisingly well. We're still waiting for a major roadblock. I think that is Antep at one point. But we've really progressed amazingly quickly. When we founded the company, we first had to upscale our screening platform onto a robotic setting to actually screen in high throughput. And now we're about to enter mouse studies next month, basically. We have a second program that we licensed in last year, also from UK of Zurich. And there, it's even faster. We started it later, and we're also about to enter mouse studies.
Okay, well, and I guess if you do it second time, it's faster.
Also, yeah, absolutely.
On the robotic is an interesting point. So you really built your whole robotic platform, or did you reuse existing things, and why did you outsource it or delegate it to someone else?
We have access to ETH technology platforms.
Okay.
So they have a robotic screening platform with a proprietary high diversity small molecule library. So we use theirs.
Okay, yeah. Because it sounds pretty, cash intense and complex.
No, I mean, what we do is cash intense anyway. So. So we screen with ETH, and medicinal chemistry, which is one of our value drivers really, is also outsourced to a company to a CRO in Holland.
Yeah, makes sense. I will come back to the ETH in the environment. On the finance side, so cash intense. I was pretty surprised when you announced the Series A. It's a really big round, especially for a company that was started 1 year ago. So how did you manage that?
Our seed round was 2.5 million Swiss francs.
Yeah.
That was in 2017, about 6 months after.
But that was a reasonable round, like announcing just after the creation.
Okay,
but then just one year after a big Series A.
The lead investor, the lead VC we had on the round, Biomet Partners, they've really been following us since foundation. And I think what they wanted to see is whether we can really deliver what we promise. And we did, and they were super happy. So that's why they decided to invest.
Makes sense. No, that's good. So maybe on the team, I saw you were looking for a COO. So you found him now, him or her?
We have a couple of very, very interesting candidates. Yes.
Okay. I guess it's challenging to find. This position, there's not that many good CEOs in Europe actually.
Yeah, it's extremely challenging to find the right profile. We got over 200 applications for that position. 200 CVs after screen. And a lot of them are interestingly big pharma people that are somehow attracted by, a little startup where you can actually build something.
Yeah.
To us, these were not extremely interesting candidates.
Yeah.
But we found some really interesting biotech people.
That's good. You need the right fit. You are on the active in the leadership. You're the only founder active.
I am, yes.
Yeah, so then it's even more important.
Absolutely, it can't stay a one-woman show forever.
Definitely. And so, how big is the team at the moment?
We're 7 people internally, and we have 10 full-time medicinal chemists at the CRO.
Yeah.
The project leader and a very, very active consultant for medicinal chemistry.
Yeah, sounds good. And I guess you will hire a few more with following the round. Yeah. Great. And maybe so besides signing the CRO, what's your biggest challenge at the moment? Or challenges?
Biggest challenge, I mean, certainly finding that COO is really, really key. That's not, it's difficult to judge people just from meeting them a couple of times and giving them case studies.
Yeah.
That's certainly a challenge. Probably the biggest one. We have founders and our advisors. We have a lot of expertise at the table already.
Yeah.
Fundraising is fine so far. It's really building a strong team.
It's crazy, and hiring. I mean, I hear it a lot from executives or founders that they really struggle to hire, especially like more senior people. Do you like? I mean, I guess you look Europe-wide or even globally, like US as well, or do you focus like on more local?
We've gotten applications from the States as well.
Yeah. Wow.
But our prime candidates are based in Switzerland.
Yeah. I think that's a good thing with Switzerland. You have a lot of expertise in biotech/pharma/life sciences, and it's actually Thomas mentioned that from Pharma that he has access to really good talents, local talents, well, at Swiss rate, but experienced. All right, and so until the end of the year, like the milestones you expect to reach?
We would like to have, for our 2 assets that we're developing, a preclinical proof of concept, so that we know our compounds can really prevent the development of drug resistance in relevant cancer models.
Yes. Sounds like a good milestone already.
Pretty important milestone to have.
Yeah, definitely. Good as that one. So maybe now let's just switch a bit and take a bit of perspective. Just asking your feeling on the Swiss biotech ecosystem, a founder ecosystem. It seems like at least the VC side treats you well. I mean, you have Biomed Partners mentioned and Wedelpin as well, which I think are the two biggest biotech VC with BB Biotech. But so how, besides the VC, what's your feeling on the biotech system in Switzerland? How did it impact the company? How did it help you?
In terms of funding, you mean?
In terms of the whole company, or how much did it contribute to where you're at right now?
I would say it contributed a lot, especially getting us off the ground. Our seed round was covered by mainly high net worth individuals that almost all of them have some sort of a biotech background. And I think that certainly helped. You really have players at the table that know what they're talking about, and it gives you a certain validation as well. Just the understanding of the process as well. They know what drug development is, the risks that are associated with it. And I think that is important, that we all have the same idea of what we're getting into together. And I think personnel-wise, it also helps that we have a strong pharma and biotech culture in the country. Also, some of our people that work for Tolremo now used to work at Roche, they used to work at Molecular Partners, so we're really able to profit from that as well. And
I guess from ETH as well, with access to infrastructure and
absolutely, that is key.
That's really brilliant from ETH. It's not that common in at least in Europe. I don't like. I mean, some universities do that, some kind of accelerator and kind of some shared things, but usually it's always. And I never hear really good feedback from people who are inside of it, which usually is not a good sign. But from ETH, I hear, or even Lausanne as well, ETH in Lausanne, I hear very good feedback. So,
yeah,
which I think is a good sign. Okay, great. And my second last question was on, so that how is it in the biotech industry as a young female founder or young female founder? How is it to be in that industry, which is very more mature and very masculine industry? How is that?
I don't think it's different for women than it is for men. I
don't know.
But I never felt that it was an advantage or a disadvantage. It's just how it is.
I think that's the best. Just go for skills and prove that that's good what you're doing and get some data and then.
Exactly.
Okay.
We're all in the same boat there.
That's good. I think that's how we have this. We have this culture internally as well. And that's how we hire. We don't really care. It's if the person is good and it's a perfect fit, we hire the person. Sounds good. If you want to expand on it, it was a very short answer. But,
it's one of those questions that you always get as
it's a touchy topic, but it's an important topic, especially in biotech. I think
I think over time it will probably become less and less important. When you have more girls going to high school, more girls starting to study STEM subjects, naturally we'll have more young women founding companies and joining boards. And I think in a couple of years, hopefully, it won't be that much of an issue anymore.
It'll just take time. I agree. All right, and my last question is who should we interview next?
Yeah. I would say, since we talked about women, maybe Olga Dubé, I think her name is, AgroSustain.
I didn't hear the company. So Olga from?
AgroSustain.
AgroSustain, okay.
They make, they basically prevent food from going bad.
Yeah.
It's a couple $1 billion market. They have a small compound that can spray on food and prevent your strawberries from getting all moldy within a couple of days.
That sounds good. Yeah. I don't know her. I will check her out and let you know.
Very cool.
Cool. Well, thanks. We're just on time.
Just on time.
We have 30 seconds left, which is good. I can say.
That's what happens when you talk to smooth entrepreneurs.
Trains run on time.
Exactly.
That's great. Well, thanks a lot, Stefanie, for the chat, and good luck with all your challenges and keep up.
Thank you.
Yeah, it's great. Thanks.
Thank you. All right.
Bye-bye.
Bye-bye.